A good morning or afternoon to you. Thank you for joining us. I'm Jan Riker, Chief Operating Officer of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics relating to antibody discovery and development. Our expert speaker, Dr. Lenny Moise, will discuss strategies for mitigating the unpredictability of Fc-mediated functions in antibody development. Please note the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I now turn the show over to our speaker.
Thank you, Janice. Thank you to the Antibody Society for this opportunity to present. I hope this webinar will get you all thinking about the interconnected factors that influence antibody effector functions and how antibody designers and developers like you can manage this complexity and unpredictability by using a high throughput approach to comprehensively characterize Fc effector activity. At SeromYx, we focus on measuring functions of antibodies that are mediated by the Fc and how these functions can be used to develop safer and more effective therapeutic antibodies. In addition to their well-known role directly neutralizing viruses or binding therapeutic targets, antibodies mediate a wide variety of functions by harnessing the potential of the innate immune system. Most of these functions are not examined today during the antibody development process. So what am I talking about? What are these functions? Surface expressed antigens are recognized by antibodies, which function as molecular beacons, marking the antibody target, be it a tumor cell, an infected cell, or a pathogen, marking them as aberrant and providing instructions to recruited innate immune cells on how to best eliminate the target, including release of pro-inflammatory cytokines, phagocytosis of target cells and pathogens, direct lysis by NK cells, degranulation of granulocytes, activation of the classical complement cascade, and inducing the activation and maturation of dendritic cells to drive more effective T-cell immunity. There's a lot going on here, and this illustration isn't comprehensive. Yet, most functions are not examined today during the antibody development process. And it's not because the assays don't exist. There are robust assays to evaluate many antibody Fc effector functional and biophysical properties. The common practice is to characterize a small number of functions, and these are typically driven by requirements that are imposed by regulatory authorities. The typical assays are antibody-dependent cellular cytotoxicity, or ADCC, which is mediated by NK cells, complement-dependent cytotoxicity, CDC, and sometimes antibody-dependent cellular phagocytosis, or ADCP, mediated by monocytes. But the array of functions is much broader than this and may well be relevant to therapeutic safety and efficacy. For example, you might want to know if your antibody is capable of engaging Fc receptors on neutrophils to mediate cancer cell elimination by ADCC versus the ADCC that's mediated by NK cells. Mouse lymphoma studies have shown that depletion of neutrophils reduced the efficacy of alemtuzumab, which is directed against CD52, and rituximab, which targets CD20. You might also want to know if your cancer immunotherapy provides a direct link between innate and adaptive immunity through dendritic cell or neutrophil endocytosis of Fc gamma receptor bound immune complexes that can drive antigen presentation, activation of cytolytic CD8 T cells that recognize cancer antigens, and development of cancer-specific immune memory. In terms of safety, You might want to know if your antibody induces cytokine release syndrome via an Fc receptor dependent mechanism. Cytokine release syndrome has been described after infusion of several antibody-based therapies. It's a systemic inflammatory response with a range of clinical effects from fever and fatigue to multi-organ failure, making it a safety risk that's important to anticipate. So these are just a few examples why it is important to more broadly characterize antibody Fc effector functions. We at SeromYx broadly characterize effector functions using a suite of biophysical and functional assays. The platform was spun out of Galit Alter's lab at the Reagan Institute of Mass General Hospital, MIT, and Harvard, and it was industrialized by SeromYx to support antibody developer needs from discovery through licensure. So what do I mean by industrialized? There are several points. Let's start with breadth. For therapeutic antibody characterization, we use biophysical assays that assess glycosylation profiles and Fc binding to receptors, including an array of Fc gamma receptors, Fc alpha receptor, neonatal Fc receptor, FcRn, and C1q. Functional assays go beyond the standard ADCC, CDC, and ADCP and include phagocytosis mediated by neutrophils, dendritic cells, eosinophils, and basophils, as well as NK cell activation, complement deposition, and mucin binding. But breadth is just where we start. There are several more points to consider in characterizing antibody functions. A key point is whether assays are run in the presence or absence of antigen. I think this is underappreciated sometimes. It may sound obvious that effector functions occur in the context of antigen binding, but it isn't uncommon to find Fc binding studies that are conducted in the absence of antigen, and then that is used to set expectations for effector functions that are measured in the presence of antigen. I will speak to this point more a little later, but for now, know that every assay in our platform is antigen specific. Next, it's important to consider how closely an assay recapitulates functions in vivo. Some cell-based assays use reporter readouts that tell you an immune complex bound an Fc receptor and initiated a signaling event, but they don't measure the very functions that contribute to product efficacy, such as cell killing and phagocytosis. We believe it's important to know that an antibody product can induce these functions if the product is designed for that purpose, or to know that it doesn't induce those functions if the goal is to silence effector function. The bottom line is measure the bottom line. Most of our work today is done to support regulatory findings or filings. So it's difficult to overemphasize how important it is to implement a quality management system. Why? To ensure consistency, reliability, compliance, while at the same time reducing risks, enhancing efficiency, and maintaining high standards of quality. A quality management system is the basis for how we operate our lab under GCLP compliance to generate high quality data that is reliable and reproducible while adhering to regulatory standards. Then there's experience, and there's no substitute for experience. We have broadly characterized hundreds of candidates across multiple functions, performing tens of thousands of assays with diverse clients in government, global health organizations, pharma, and biotech. And the final piece in the platform is cost effectiveness. Thinking big picture, in the drug development process, it's better to identify failures and reduce uncertainty earlier than later. Broad characterization of effector function early in development is a cost-effective measure that can reduce the risk of unexpected findings after advanced preclinical development of a lead candidate, or worse, in clinical trial. So these are the bedrock features of our platform that we take into the challenging world of understanding, optimizing, and predicting function. So why is it so challenging? Simply because there are a multitude of interconnected factors that influence antibody effector functions. First, and you all know this, the properties of the Fc architecture itself impact effector function. Variations in antibody isotypes and IgG and IgA subclasses encode variable Fc interactions with Fc receptors that have consequences on stimulation and inhibition of effector functions, complement stimulation, and serum half-life. I should also mention that isotypes and subclasses also encode variation in hinge length and flexibility that modulate effector function. It's also well known that variations in antibody glycosylation patterns impact antibody structure, flexibility, and affinity for Fc receptors, and consequently, effector function. As I alluded to earlier, there are properties of the Fab in forming immune complexes that have significant impact on function. Now, it may not seem intuitive that what goes on one side of an antibody could be dependent on what happens on the other, but there are a number of features involving the Fab that strongly impact Fc-mediated functions. Affinity for the target antigen and valency impact antibody density that's needed to reach the threshold for activation of effector function. Another factor is epitope specificity and binding geometry. That is for antibodies that recognize the same antigen. For example, in a study using a model of antibody opsonized target cells, antibodies that were greater than 10 nanometers from the target surface showed significantly reduced ADCP activity. In contrast, epitopes closer to the surface excluded inhibitory CD45 phosphatase, enabling Fc receptor ITAM phosphorylation and leading to phagocytosis. The interaction of the Fab with antigen may also induce long-range conformational changes that reach the Fc, affecting its ability to interact with Fc gamma receptors. This is referred to as Fab-Fc conformational allostery. Antibody allostery is a new model of antibody Fc receptor interactions that competes with the traditional associative model that assumes that Fc receptors don't discriminate between IgG antigen complexes and free IgG. Whatever the relative roles of the associative and allosteric interactions is and remains to be clarified, the building evidence that supports conformational allostery should be considered in the big picture of the interrelated factors that impact Fc-mediated effector function. The fact that the Fab domain impacts function suggests that the process of selecting a Fab and an Fc independently is fundamentally unsound. The final piece in the pie is the effects of improving on nature. Antibody engineering is motivated for a long list of reasons. Many of them are shown here. Most work is done on the Fab, obviously not aimed to impact Fc-mediated functions, but nonetheless can result in changes that are sometimes unexpected. These engineering aims include increasing target affinity, increasing the number of epitopes that are recognized, and that has led to development of multiparatopic and multispecific antibodies, and increasing the number of potential binding sites, such as development of bispecific and bivalent antibodies. Of course, engineering the Fc is intended to change effector function, either increase one or more functions or silence function where it's not required or desired. when many studies have shown that IgG function is enhanced by point mutations at the Fc receptor binding site and the overlapping complement binding site, or by glycoengineering. For example, afucosylation is known to increase the affinity of IgG monoclonals for Fc gamma receptor 3A on NK cells, driving greater cell killing by ADCC. I want to highlight a couple of recent innovations in Fc engineering. One is introducing point mutations at conserved glutamate positions in the Fc. These mutations promote Fc-Fc interactions that induce hexamerization and effectively lower the threshold for triggering complement activity compared to conventional IgGs. The other innovative approach isn't meant to dial up or to dial down effector function, but rather it fine-tunes Fc activity to the tumor microenvironment for reducing on-target off-tumor side effects through pH-selective mutations in the Fc. While these site-specific mutations can enhance product efficacy, safety, they may affect antibody conformation at distant sites, resulting in changes to effector function. Similarly, half-life extension mutations that are introduced into many antibodies that are in development, they don't aim to impact effector function, but may have unintended structural effects that could impact therapeutic efficacy. It's the interplay of these various antibody and immune complex effects that makes for a delicate balancing act when engineering antibodies. In the coming slides, I'll give examples how these efforts to improve on nature can have unexpected effects on effector function. Let's take a look at an example of Fab engineering. Increasing antibody affinity and specificity is a critical objective in development. However, effector function could be a trade-off, and if effector function is essential for efficacy, it would be counterproductive to affinity mature at some point. This is illustrated in a couple of studies out of AstraZeneca that show higher antigen binding affinity does not always correlate with increased Fc effector function. These studies assessed antibodies that target EGFR and HER2. Antibodies with moderate affinity showed higher levels of CDC, ADCC, and ADCP when compared to high affinity variants. Now, this is an unexpected finding. How is it explained? Well, it's thought to be that shorter off rates for antibodies with higher affinity are associated with greater bivalent binding, leading to reduced density of Fc domains on the target cell surface. Lower densities lead to a lower functional response. Conversely, antibodies that have faster dissociation rates could show higher levels of monovalent binding that would facilitate higher levels of cell surface opsonization, and higher densities support higher effector function. The take-home message is the requirements for both high antigen binding affinity and robust effector function underscores the need for screening approaches that can effectively guide the selection of candidates that don't sacrifice effector function. Let's now turn to examples in Fc engineering. An important consideration is the balance of antibody binding to activating versus inhibitory Fc gamma receptors. An Fc that demonstrates high affinity for activating receptors and lower affinity for inhibitory Fc gamma receptor 2b could yield optimal effector function. However, Fc binding sites for activating and inhibitory Fc gamma receptors are practically indistinguishable. So this poses a significant challenge in Fc engineering, even when using structure-guided computational strategies. The net result could be that modifying the Fc to target specific Fc gamma receptors can inadvertently impact binding to Fc gamma receptors and product efficacy. For example, a Xencor study that aimed to enhance anti-HER2 trastuzumab ADCC function by improving Fc gamma receptor 3A binding found that variants with the greatest enhancements in Fc gamma receptor 3A affinity also significantly increased the binding to Fc gamma receptor 2B. Increased Fc gamma receptor 2B binding presents a significant risk to efficacy, as has been shown in animal models. For example, trastuzumab-treated tumor-bearing mice that lack Fc gamma receptor 2B show increased ADCC, suggesting that Fc gamma receptor 2B is involved in inhibition of ADCC. Conversely, mice that were deficient in activating Fc receptors don't control tumor growth. So modifications that increase Fc gamma receptor 2B binding have the potential to result in greater inhibition. In a similar study, but using an EFTAE Fc variant of an anti-CD20 antibody, which was designed to increase Fc gamma receptor 2A binding, indeed enhanced binding to Fc gamma receptor 2A. However, it did not result in improved phagocytosis, which was attributed to augmented binding to inhibitory Fc gamma receptor 2B. Taken all together, the requirement for precise specificity and strong affinity for Fc gamma receptor subsets and the unpredictability of mutational effects underscore the need for screening approaches that can effectively guide the optimization of Fc domains that are tailored to various therapeutic applications. Another key point is that even when Fc modifications achieve the desired effects on effector function for one antibody, there is no guarantee that the same modifications will have an identical effect on a different antibody. So let's turn back to that EFTAE Fc variant. So these mutations near the Fc gamma receptor and C1q binding sites can significantly increase CDC, as was shown for monoclonals against CD20 and CD40. These mutations even imparted CDC function to anti-CD19, which lacked CDC activity in its original form. However, CDC potency and efficacy was not identical across the board. CDC is dependent on high opsonization density, and because antigen density is variable, significant differences in activity for the same modification could be observed across different targets. The effect of the same Fc modifications on the affinity of different antibodies for Fc gamma receptors is also unpredictable. For example, the silencing FES triple mutation, sometimes referred to as TM, which is present in various approved monoclonal antibody products, significantly decreases binding to Fc gamma receptor 1, 2A, 3A, and C1q for an anti-CD19 IgG1. However, Fc gamma receptor 2A, 3A, and C1q binding was preserved for an anti-TNF IgG1, albeit at a diminished level, but still preserved. So again, this underscores the importance of measuring outcomes. Turning now to half-life extension, Fc modifications that extend half-life have several advantages, most notably for the patient. Longer half-life means less frequent dosing, which is a significant advantage for patients that have chronic conditions that require repeated administrations of the antibody treatment. Fewer treatments and healthcare visits can also lead to cost savings for healthcare systems and patients. However, half-life extension would be counterproductive if it comes at the expense of effector function, as illustrated here with data from our own lab. This is only one example. We have seen this a number of times. For example, in published work we did with Ology Biosciences that is shown here for 2 monoclonal antibodies that target SARS-CoV-2 spike, we found the 3 amino acid YTE modification increases FcRn binding for each antibody, as you would expect. However, the YTE modification reduced ADCC, NK cell activation as measured by surface CD107A and intracellular interferon gamma, as well as neutrophil-mediated phagocytosis and complement deposition. Once again, the unpredictability of mutational effects drives home the need for screening approaches that can effectively guide the optimization of antibody effector functions. So that is exactly what we do. We measure outcome. We can speak to the impact of different antibody designs on effector function. For example, what is shown here is the maximal ADCP activity we measured for a set of about 600 different monoclonals that bind the same antigen. The Fabs are different, but the Fc is fixed, meaning epitope specificity and affinity are variables, but the Fc is not. So as you can see here, some antibodies are functionally inert. These are the antibodies with phagocytic activity between the yellow and green dotted lines, representing the no antibody and irrelevant monoclonal antibody controls. The red bar highlights that for a given concentration, the range of maximal phagocytic activity is large, even though all these antibodies share the same Fc. So testing is required to find antibodies with high function, and even then, the antibody concentration that supports high function can vary widely, as illustrated by data in the green box. This is true for other antibodies as well. We have found the same effect for a different set of monoclonals that have an identical Fc but different Fabs, as shown here on the right. What we found is the diversity of function is not only seen for monocyte-mediated phagocytosis, but also neutrophil phagocytosis, ADCC, NK cell activation, and complement deposition. Let's turn now to the converse case. Now the Fc is variable and the Fab is fixed. So let's say you've identified a Fab, and now you want to tailor effector function to meet your target product profile by screening a library of Fc variants. In the case study shown here, Bonnie Gunn and colleagues had screened a library of over 60 Fc variants on a single Fab and assessed monocyte and neutrophil-mediated phagocytosis, NK cell activation, and complement deposition. The petal plots here show the various ways that the antibodies were classified based on their functions. In comparison to the wild-type IgG1, which showed only ADCP activity, and a functionally silenced Fc variant, the antibodies showed a high NK cell function profile, or high phagocytosis and complement activity, or a balanced mix of functions. So what is the optimal set of functions? The answer was determined using animal model outcomes to link to the Fc screen. Animals that were treated with Fc variants that support high NK cell function had clinical outcomes similar to the wild-type IgG1 and non-functional antibodies. In contrast, the high phagocytosis and complement and balanced function monoclonal antibodies showed protection against disease. So this study illustrates not only the possibility of broadly screening for effector function to guide antibody design, but also the importance of identifying the desired functional profile for product efficacy. The tools that are needed to get this done exist. There are the industry standard assays that are a good start, but as I've shown, it's important to look more broadly. We cover more of the waterfront with 5 additional functional assays, as well as biophysical assays that characterize Fc binding with an array of receptors, and not shown here, glycan profiling. To be frank, there are more Fc receptors to cover and more functions to cover. Our aim is to build our platform to even more broadly characterize effector functions, and we'll do that in response to client demand. We constantly work with clients to add assays. And in the 4 years of our existence, we have added 3 or 4 assays and enhanced existing assays. So we'll develop those new assays the way we develop the existing assays by focusing on these key attributes. Starting with robustness, our assays are extensively developed with optimization of each assay component. As an example, for a bead-based assay, we consider bead size, lot variability, antigen coupling chemistry, and number of beads per test. With regard to antigen, we consider the antigen to bead ratio, wash conditions, antigen stability, and coupled bead stability, and the list goes on and on. Next, assays are developed to the GCLP standard to assure accurate and reliable assessments of effector functions. Some quality attributes are shown here, starting with precision, which speaks to the ability of an assay to produce consistent, reproducible results when repeated under the same conditions. We have extensive experience in qualifying assays. We have found in qualified biophysical assays, the coefficients of variations range from 10 to 15%. For functional assays, the CV is 8 to 25%. In bead or cell line based assays, and the CV ranges from 16 to 40% in primary cell-based assays. Next, linearity is a critical quality attribute which addresses the relationship between the measured response and the concentration of the analyte over a specified range of concentrations. And of course, we address specificity, the capacity of an assay to measure only an analyte or activity and not interfere with other components in the sample, and sensitivity being the assay's ability to detect low levels of analyte or activity, assuring that even small changes or differences can be observed. Our assays are adaptable. They've been adapted to a variety of antigen formats. So far, we didn't find an antigen we can't work with. Although our experience has shown that antigen could be a major source of assay variability. So identifying a high quality antigen is needed for high quality assay data. Our assays are also adaptable to various sample matrices. Of course, we work with purified synthetic antibodies, but we can also, and we actually have, characterized activity of therapeutic antibodies relative to background functions in serum collected from patients who were administered the drug. The last point is we design assays to be high throughput. Our focus has been developing this platform on maximizing efficiency and in turn throughput, while at the very same time maintaining quality. We can analyze thousands of samples in a single experimental run, running these large-scale runs many times a week. And we are with you throughout the development process. We have done or are running projects at the lead selection, clinical development, and regulatory phases. We believe it's also important to screen early to discover Fab-Fc combinations with a desired mix of effector functions and reduce the need for late-stage antibody engineering. Our platform today does not screen millions of candidates like can be done for Fab screening, but if you can narrow down the possibilities to tens of thousands, you could efficiently run hit-to-lead screening for effector function and reduce the risk of unexpected findings later in the development process. So to link back to where we started, Fc function depends on a large number of factors that interact in complex ways. Broadly measuring outcome against a large set of candidates is the best way to ensure you maximize the function you need for efficacy and minimize the function you don't for safety. And with that, thank you for your attention.
Thank you very much. That was indeed very educational. And we do have a few questions. I will ask the longest one first. I do appreciate that this attendee took the time to write out context, so I will read it in its entirety. Thank you for the insightful discussion, Dr. Moise. I appreciate the wealth of information you provided. I have a specific inquiry regarding the design of bispecific antibodies, particularly those targeting anti-CD3, anti-HER2, presumably, possibly 3, in cancer therapy. Our primary objective is to activate T cells in proximity to cancer cells. However, there's a concern about inadvertently activating peripheral T cells that may not contribute to the anti-cancer response. How do you propose we effectively control the activation of T cells outside the tumor microenvironment in order to optimize the therapeutic impact?
Well, I see that is a big challenge, and to be honest, we are not antibody designers. As I said, we measure outcomes, and we think it's important to screen. There is a study that considered a bispecific antibody, and I'm not recalling exactly which one it was, but bispecific antibody, and comparing that to a bispecific bivalent antibody. The bispecific bivalent demonstrated more on-target, off-tumor effects than the bispecific. And even the bispecific off-target or off-tumor effects were observed more so for one of the Fabs that had higher affinity than lower affinity. So I think that, not being an antibody developer myself, I think that you need to consider these various antibody design formats in the context of different affinities. So titrating the affinities to find the optimal design that will maximize selectivity for the on-tumor effect and avoid off-tumor effects. And to do that is going to require screening. So looking for those outcomes is essential in order to get to where you want to go.
All right, thank you very much. The next question, what are your assay performance challenges?
Well, as I mentioned, antigen quality is very important, and we have a lot of experience running assays for different applications, not just for monoclonal antibody development, but also vaccine development. And in all cases, we find that antigen quality is essential. But what we found in the monoclonal space, which is different from the vaccine space, is that where the vaccine antigens or working with polyclonal samples is more forgiving of how the antigen is coupled to beads for those assays we run with our with the bead technology. So how the target epitope, which is, and there's only one in a monoclonal antibody scenario, how that epitope is presented on the bead is an important consideration. And sometimes that requires screening different coupling chemistries to find the optimal presentation of that epitope that can be detected for successful assay outcomes. So that is a consideration that is something that we look at for every project, because every target antigen is different, and where that monoclonal binds on that antigen is usually unknown to us, even if it's known to the developer.
Okay, thank you for that response. One more question here, and that is, can machine learning be used to predict Fc function for computational antibody design?
Yeah, that is something that interests us because we think that despite the fact that there has been extensive mutational screening of the Fc, which has yielded a wealth of knowledge about what types of modifications to dial up or dial down effector function and or extend half-life, etc. A lot of that work is done on just a handful of antibodies, and of course that's challenging. But as I had mentioned, the impact of Fab on effector function is something that hasn't been explored. So our information or available today about the impact of changes in the Fc on Fc effector function is not as well developed as, say, how we study the Fab. So there's a lot more data in that space and a lot of work that is going on to develop machine learning approaches to predict binding. But there isn't as much work going on in the Fc space. That's something where we would like to see more development. So we get to the point where we're able to computationally design antibodies accounting for effector function with more confidence.
Okay, thanks very much. The takeaway for me on that was that we're not there yet. Not there yet with machine learning for predicting Fc function. But hopefully sometime in the future, there'll be enough data available that that can be so. With that, we do have a thank you. So thank you to the attendees for offering thanks to Dr. Moise. Great talk. I agree with that. Thank you very much. If there are no additional questions, I'll stretch that out for just a moment in case one pops in. We are now at the end of the questions. So I will say, in concluding, I do want to thank Dr. Moise for indeed providing a very insightful presentation here into mitigating the unpredictability of Fc-mediated functions in antibody development. And I thank all the attendees for joining the webinar today. An on-demand version will be available very soon. I'll send a link to it by email to everyone who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.